All right, everybody, welcome to part one lecture on the brain. In this part, I'm going to talk about some different areas of the brain, and we're going to start with development. So first, I want to bring up the idea of cephalization, and essentially all cephalization is referring to the evolutionary development of basically this anterior part of the brain, the also called the rostral portion of the brain. And because of this development, this has resulted in an increased number of neurons. As we can see, the highest level of cephalization actually is reached in the human brain. And that's sort of highlighted here in this picture to the right, where you can see the telencephalon of the human brain, this most anterior and rostral portion is incredibly large, and this gives humans what we call human-like properties like executive decision-making, impulse control, and so on. So basically, this term of cephalization just refers to the idea that many creatures have evolved to having a head where most of the sense organs have become concentrated, like our eyes, ears, mouth, nose, and tongue. So let's go ahead and let's discuss embryonic development. So the brain and the spinal cord form from what is called the neural tube, and that's what you see over here on the right. And the nervous system in general is one of the very first structures that begins to develop during embryonic development. But basically what happens is this neural tube, it expands. The anterior side expands, and that's going to form three what we call primary vesicles. And those three primary vesicles include the prosencephalon, which is the forebrain, mesencephalon, which is the midbrain, and the rhombencephalon, which is the hindbrain. So basically, you can see on this picture three distinct areas. So if I drew little circles around them, there's 1, 2, 3. All right, and so each of these three different areas have developed from this anterior portion of the neural tube. So the prosencephalon, mesencephalon, and the rhombencephalon. Now this posterior, this caudal portion of the neural tube actually goes on to become the spinal cord. And whenever I'm talking about the vesicles, I just want to also point out that these are sort of hollowed out structures. So the blue interior portion is going to be filled with fluid, and it's more or less the outer portions that are becoming tissue. All right. So from here, we can see that the primary vesicles are going to give rise to secondary vesicles. Okay, so for example, the prosencephalon or the forebrain is going to give rise to this telencephalon and diencephalon. The mesencephalon is going to give rise to, well, it's actually going to stay the mesencephalon. And then the hindbrain, the rhombencephalon, is actually going to further divide into the metencephalon and the myencephalon. But we're not quite done yet, because each of these secondary brain vesicles that we just talked about are going to go on to become different brain structures in the adult. So the telencephalon, you can see this sort of tan colored outer region here, is going to go on to become the cerebrum. The diencephalon, which is outlined by this darker blue area, is going to go on to become the thalamus, hypothalamus, epithalamus, and also the retina of the eye. The mesencephalon goes on to become the midbrain portion of the brainstem. The metencephalon actually is going to become two different regions. So you can see This sort of orange outer tissue layer is going to become the cerebellum, and then this sort of green inner tissue layer is going to become the pons of the brainstem. And then finally, this myelencephalon is going to go on to become the medulla oblongata of the brainstem. And then again, all of the inferior portion of the neural tube is going to go on to become the spinal cord. And remember I said it's this outer tissue layer that's important, that's going to become the tissues, it's going to become the neural tissue. And then this inner layer was kind of hollowed out. This is because this inner layer is filled with fluid called cerebrospinal fluid. And the cerebrospinal fluid basically is going to be located in this area in what are called ventricles. So inside of the telencephalon is where we're going to end up finding the lateral ventricles. Inside the diencephalon is where we form our third ventricle. Mesencephalon, we're going to form what's called the cerebral aqueduct. The fourth ventricle is going to be formed by the combination of the metencephalon and myelencephalon. And then finally, the spinal cord, the region of the spinal cord that's going to contain circulating cerebrospinal fluid is called the central canal. And so this is just a blown up picture of everything that I just talked about, starting from the neural tube and progressing into its adult brain structures and adult neural canal regions. Now, what's interesting is that because the brain actually grows faster than the skull that's surrounding it. And at this point, the skull would be membranous. It's not fully bone yet. So this is sort of a membrane sort of situation. The brain itself is actually growing faster. And so it actually has to fold in order to occupy the space that's available for it. And so whenever it folds, we're going to have the forebrain actually moving. So this is all forebrain is actually going to be moving towards the brain stem. And then we also have the cerebral hemispheres, which is also going to be sort of part of the forebrain in a sense, is actually going to here in this tan color, it's going to sort of double back and it's going to basically engulf this purple area, which is what you see happening down in this bottom structure, where you can see that at week 13, the cerebral hemispheres have grown to enclose the diencephalon and the rostral portion of the brain stem. Okay, so as it's doing this, this creates creases and folds, which increase the surface area that's available for neuronal development. So I just want to briefly touch on what are called neural tube defects. These are defects in the brain, the spine, or the spinal cord that occur usually during the first month of pregnancy. And most of the time these occur before a woman even knows that she's pregnant, but most cases of neural tube defects are caused by a lack of what's called folic acid. Okay, and so you might have heard of folic acid. It's a supplement that some people take regularly, but it's highly recommended for pregnant women. And this is just to decrease the likelihood of your baby developing a neural tube defect. Because the United States has made the recommendation to supplement pregnant women with folic acid, the incidence of neural tube defects has significantly decreased. And so neural tube defects are essentially broken down into two major categories. So these neural tube defects can either be anterior or they can be posterior. So if they're anterior, we're talking about a brain defect. If it's posterior, then we're going to be talking about a spinal cord defect. Okay, so first we're going to talk about this anterior category, specifically anencephaly, which is what you have over here, this picture. This is the most severe anterior neural tube defect that results in spontaneous abortion because at this point the baby is developed without a brain and there are massive defects in the skull, the meninges, and the scalp. Okay, so this is like worst case scenario. Alternatively, we have posterior defects, and remember I said posterior defects are defects that are occurring in the spinal cord. And so what you see happening here, so I just included a picture of the normal vertebra, what it would look like. And if you remember from lab, we have this open hollow space that we call the, the central canal of the vertebra. And this, remember, is where we said that the spinal cord is going to be located. All right, and so here you see a vertebra with the spinal cord located inside of that area. In these posterior defects, there's usually just something didn't go right here. So in the first case example, spina bifida oculata, we have a case where the vertebra itself did not fuse all the way. Okay, so we actually end up not having a spinous process here. You can see that there. And usually this is no big deal. Very rarely does this require any sort of surgical fixation. And interestingly, what usually happens is that area will be present with a tuft of hair. And so that's a common way that it's recognized. Once the baby is born, the physician will notice a tuft of hair in that area, at which point you might suspect spina bifida oculata. The next type of defect, posterior defect, is called spina bifida cystica. And spina bifida cystica is slightly different in that you can see there's no vertebral arch. So normally the vertebral arch would be in that area. Okay, so we have an incomplete vertebral arch. Additionally, what's happened is that the protective surroundings of the spinal cord are, two of them are completely herniated to a pouch outside of what would have normally been this area. So it kind of looks like a giant cyst on the baby's back. The spinal cord, it still is pushed up against the vertebra. Spinal cord is still in place, but what happened is that the protective coverings around the spinal cord, called the dura mater and the arachnoid mater, are actually protruding outwards into this sort of pouch. Okay. Finally, the most severe type of posterior defect is called myelomeningocele, and this is a complete displacement of the spinal cord to a membranous sac outside of the vertebral canal, the vertebral canal here, this big hole in the middle of the vertebra that should be containing the spinal cord. At this point, it's similar to spina bifida cystica, the difference being that in myelomeningocele, the entire spinal cord has been displaced. Okay, and so this is what this would look like on a baby, and this would definitely require surgical fixation to put the spinal cord back in place. So now that we've talked about how the brain develops, at birth, the brain is going to start showing its adult pattern of organization. And so we're going to have major regions here, including the cerebral hemispheres, the diencephalon, which you see in purple, the brainstem, which is going to have 3 different regions, and then also the cerebellum. So before we talk too much about that, and here's just another breakdown of those, of how the adult brain is arranged. But before we talk too much about each of those different regions, I just want to point out that since we talked about nervous tissue last week, the central nervous system does have both gray and white matter, and the gray matter is going to house the cell bodies of neurons and also axons that are really short that don't have myelin on them. However, the white matter is going to be mostly composed of myelinated axons, though there might be some non-myelinated axons, but for the most part, it's myelinated axons. And that's actually the whole reason we call it white matter, is because myelin to the eye appears white. And so the presence of myelin gives the tissue in that location a white appearance, which is why we call the area where there's myelinated axons the white matter. So the basic pattern of the central nervous system is that we have a central cavity, which is going to have cerebrospinal fluid. And then just around the central cavity, we're going to have gray matter, which is what you see as this like darker colored tissue. And then just outside the gray matter, we're going to find white matter. Okay. This is a general pattern. So in the spinal cord, so we're going to start down here in the spinal cord, it has this basic pattern. Okay, so we're going to have this pattern where, again, we have the central cavity, the inner gray matter, and the outer white matter. In the brainstem, however, things start to change a little bit. So we have, in the brainstem, we can see we have our central cavity here, and we can see our inner gray matter, and we can see our white matter out here, but we can also see other areas of gray matter. Okay, and so these other areas of gray matter are actually called nuclei. Right, and so again, this is at the level of the brainstem. Now, as we transition up to the cerebral hemispheres, also specifically the cerebrum and the cerebellum, the cerebrum and the cerebellum are going to have an outer layer of gray matter. Okay, so outer layer of gray matter, and this is going to be called the cortex. All right, so I just outlined the cortex. And then we're going to have some white matter, which is all this that you see in here, okay? However, we're also going to have scattered areas of gray matter nuclei amid this white matter. So it's kind of similar to the brainstem in that aspect. So we can see we have these sort of random, they appear random, areas of nuclei that is scattered within this white matter. I bring this up because we're about to start talking about, first of all, the cerebral hemispheres, which form the superior part of the brain and account for 83% of the brain's mass. Okay, so when I'm talking about cerebral hemispheres, I'm referring to that whole area and left hemisphere, right hemisphere. Okay, we have different sort of surface structures that when you're looking at the brain that we name. So we have gyri, which are like ridges. And so if you think about it like a hill and a valley, two hills and a valley. So we have ridges, and these ridges are the same as sulci, or excuse me, gyri. So let me just redraw this. We've got two hills. The gyri are the hills, and in between those hills is this valley, this sort of deep cave. This is a sulci. Okay, so putting that into sort of perspective in terms of looking at the surface anatomy, we can see the gyri, right? So all of these sort of squiggly things that you see are all gyri. And then you can also see in between those squiggly things, these darker lines, those are just little hills in between gyri. Okay, and so those are sulci. Right? So in addition to having sulci, sometimes in between gyri we have really, really deep grooves. Okay, so these really deep grooves. And these really deep grooves are called fissures. So there are two major fissures that I would like to point out. We have first this longitudinal fissure, and the longitudinal fissure is the one that divides the brain into left and right hemispheres. Okay, so it's a really deep groove that divides the brain. You can see that deepness of the groove here. It's dividing the brain into left and right. Okay. In addition, we also have what's called the transverse cerebral fissure. And this is a really deep groove that separates the cerebrum from the cerebellum. Okay. Okay, so I just talked about how a sulcus or sulci are the little valleys in between the gyri. And in the brain, we have several sulci that are dividing each of the hemispheres into 5 lobes. And 4 of those lobes are named after the skull bones that lie on top of them. And then one of them is named sort of completely different. So insula, this number 5 here, is just its own thing. So we have the frontal lobe, which is up here in pink. Parietal lobe is in this sort of teal blue color. The temporal lobe is that sort of deeper bluey purple color. And then we have the occipital lobe, so similar to where the occipital bone is located. And then finally, we have this insular lobe, and that's what you can see down in this bottom right picture. If you pulled back portions of the frontal lobe and also the temporal lobe, inside of the brain, deep to those, you would find the insular lobe. Okay, so down in that area is where you'd find the insular lobe. Okay. So I just said that there are major sulci that are dividing lobes. The first one is the central sulcus. The central sulcus separates what's called the precentral gyrus of the frontal lobe from the postcentral gyrus of the parietal lobe. So the precentral gyrus is located in the frontal lobe, so it would be that guy right there. The postcentral gyrus would be this guy right there. Okay, and the central sulcus is dividing those two. Okay, so you can see the central sulcus dividing those two. Okay. The parieto-occipital sulcus is going to be a sulcus that separates the occipital and the parietal lobes. And so you can actually see that back here, right, in this area. And finally, the lateral sulcus is actually going to outline the temporal lobe. And so the lateral sulcus, oops, sorry about that. Undo that. I have to go back. There we go. Now the lateral sulcus is going to be approximately in that region. And the lateral sulcus, you can see it's separating temporal from frontal, also temporal from parietal, also temporal from occipital. Okay. So each hemisphere is going to be divided into 3 basic regions. And I briefly mentioned this already earlier, but let's go back to it because we're going to focus on this specifically. So we are going to have 3 regions. First region, this outer layer called the cortex, right? Where it's just this outer gray matter area. We are going to have the white matter internally. So all of this white appearing stuff. And then we're going to have the basal nuclei, which are going to be deep within the white matter. So that's going to be these deeper areas of gray matter within the brain. Okay, so let's talk about each of these things individually. Starting with the cortex, which remember the cortex was that outer layer of superficial gray matter. So I outlined it. It was like something like that, right? Come in. Okay, so something like that. It's like a really bad drawing, so I'm gonna just erase that. The cerebral cortex is the executive suite of the brain and accounts for 40% of the brain's mass. So it's that, again, really thin superficial layer of gray matter that's made of the neuron cell bodies, the dendrites, glial cells, and blood cells. Note, very important point here. No axons are present
in the cerebral cortex. The cerebral cortex is the place where we would find functions like the conscious mind, awareness, sensory perception. This is where we initiate motor responses. There's also some communication, memory storage, and understanding going on here. And so we can see this outlined by what's called a functional MRI. And this is going to show specific motor and sensory functions are located in specific cortical areas, and these cortical areas are called domains. So for example, the domain for seeing would be located here, where the orange is. Here we can see the domain for hearing, the domain for speaking, and then finally thinking domains. You can see those are much more widespread. So there are 4 general considerations about the cerebral cortex. And general consideration number 1 is that the cortex has 3 functional areas. So areas that serve some sort of function. There are motor areas, which control voluntary movement, sensory areas, which is where we have conscious awareness of sensation, and then finally association areas, which are the areas that integrate a lot of different information coming in. Point number 2, each hemisphere is going to be concerned with the contralateral side of the body. So what does that mean? That means that if we have the left hemisphere and the right hemisphere, and I don't know, let's say okay, that's your right hand. Say you raise your right hand. That looks like a not very good drawing. I'm really sorry. Okay, you raise your right hand. That is the responsibility of the left side of the brain. Okay, so opposite sides. It's also important to understand that lateralization of certain functions only can occur in one hemisphere. And so what does this mean? This means that here's our left hemisphere, here's our right hemisphere. Let's say we're talking about, I don't know, speech. Let's talk about speech. The area, because speech is a very special function, this particular function of speech only has a special area in one hemisphere. Okay, and actually for speech tends to be located in the left brain. Alternatively, something else to kind of understand here is that any kind of conscious behavior is going to involve the entire cortex in some capacity. All right, so it's going to be an integration of many different areas in order to perform human-like things. Okay. So I just mentioned on the last slide the 3 types of functional areas. And so we're going to start talking about those now. So in terms of motor areas, we're going to have 4 major motor areas. All of them are located in the frontal lobe. Okay, so all motor areas are in the frontal lobe. So we're going to have the primary motor cortex. This is going to be located in our precentral gyrus, which, remember, the central sulcus here separates the precentral gyrus from postcentral gyrus, which was here. So primary motor in the precentral gyrus. We're going to have premotor area. We are going to have frontal eye field and Broca's area. Okay. All four of these are motor areas of the brain, specifically of the cortex. Which, remember, the cerebral cortex is that outer superficial area of gray matter. Okay. Okay. Let's talk about each of those things in a little bit more detail. The primary motor cortex is, again, located in the precentral gyrus. It contains these things called pyramidal cells, which basically are these really large neurons that allow the conscious control of really precise, skilled skeletal muscle movements. We have the pyramidal tracts. Those are actually located in the spinal cord. Those tracts are formed from the axons of the neurons whose cell bodies are located in this particular motor area of the cortex. Somatotopy refers to the fact that every muscle in the body can be mapped to a specific area on this cortex. Okay, so basically what we're doing is in our motor cortex, we are looking, here's the cortex I'm outlining, right? It's this outlined pink portion is the cortex. Each specific area of the cortex is associated with a specific muscle. For example, and remember, we do contralateral, so contralateral motor innervation of specific regions. With that being said, we have I'm just going to highlight some of this stuff for you. The tongue and swallowing can be mapped to this specific region of the cortex. Okay, so the tongue and swallowing, we are talking about that area. Let's say the thumb, it can be linked to this exact area of the cortex. Okay, thumb can be linked to that exact area of the cortex. Maybe we're talking about the, I don't know, hand. The hand is linked to this specific area of the cortex. Okay, so this is what I mean by somatotopy. And if this structure, this whole thing, if we're talking about the primary motor cortex on the left, this would actually be the right side. If this is the right side, so if this is the right side, and this is the left side, because this is anterior and that's posterior. So if this is the right hemisphere that we're talking about, all of these muscle innervations are mapped to the right hemisphere, even though they are located on the left side of the body. Okay, so we're talking about the left hand. the, I don't know, left eye, the left thumb, the left leg, knee, hip, so on and so forth, okay? Contralateral side. Fun fact, damage to the primary motor cortex will paralyze the muscles that are controlled by those areas. And so some of this damage can be due to things like a stroke or tumor injury. And the paralysis, because of the way that the brain mapping works, the paralysis, the effect of the damage, is actually going to occur on the opposite side of the body. So let's pretend that our person over here on the right, so this person here, we're going to say that this person has damage to the left hemisphere. Whatever this specific brain region is responsible for, the effects are going to show up on the right side of the body. So let's pretend that it's, I don't know, hand movement. Okay, hand movements. If there is damage to the area on the brain, would be on the left brain, that's responsible for hand movements, you would not be able to move the right hand. Okay, so there's this crisscrossing effect that occurs. However, muscle strength itself or the ability to perform certain movements might not be impaired. It's only the control over the movements. So for example, here, clear all my drawings. If there's damage to the premotor area, which controls the movement of the fingers, you would still be able to move your fingers. You just wouldn't be able to do it whenever you want. Okay, so voluntary control needed to move your fingers, say to type, would be lost if there's damage only to the primary motor cortex. Interestingly, what can happen though is that you have other premotor neurons that can actually be reprogrammed to take over the skills of the damaged neurons. And because of this is why some stroke patients can recover the function of like their, I don't know, hand if there's damage only to the primary motor cortex. They would be eventually able to regain conscious control over that particular muscle, but it would just take time because you have to retrain your brain how to do the functions you used to be able to do. So it's a really interesting concept here. All right, so I just mentioned a little bit about the premotor cortex. And so whenever I was just talking about damage to the primary motor cortex. So remember, here is the primary motor cortex. And just in front of the primary motor cortex is where you're going to find the premotor cortex. And the premotor cortex is basically the planning stage for movement. Okay, so this particular region is responsible for controlling learned repetitive or pattern motor skills. It also helps to coordinate simultaneous or sequential actions like, I don't know, walking, for example. You learn to walk once this area of the brain has made those connections of planning sequential steps. Okay, so like heel, toe, switch legs, heel, toe, switch legs, right? So the premotor cortex is the part of the brain that's responsible for this. And it also controls the actions, the voluntary actions that are dependent on sensory feedback. So let's say you're maintaining posture, right? You're sitting here and you're sitting up and you're maintaining posture. If you start to fall forward, your body, that your head movements, your particular like orientation in space would let your premotor cortex know that you're starting to lose your balance, and so your premotor cortex can then plan a movement to send to your primary motor cortex to allow you to sit back up and maintain your posture. All right, so it's this, I mean, the brain is absolutely amazing. I love talking about it, but I'm going to try to keep it as brief as possible because I know this is a really long PowerPoint for you guys. Okay, next we're going to talk about Broca's area. And this is one that I've actually already mentioned. I just didn't mention the name of it when I was talking about speech and being only located in one hemisphere, specifically the left, usually the left hemisphere. So again, Broca's area is the area of the brain that does the motor movement that's associated with speech. So Broca's area basically controls the muscles for speech production. And so it's involved in planning speech and also the motor activities that are associated with speech. So it's the action of the muscles that are making sounds. And just for reference, in case you didn't see, there's Broca's area. And then finally, the frontal eye field. The frontal eye field is located here. And the frontal eye field, basically, its job is to control voluntary eye movements like looking to the left, looking to the right, looking up, looking down. Its job is to control those voluntary eye movements. All right, so that takes care of the motor areas. Now let's talk sensory areas. All right, so we did everything in motor, and now let's talk sensory. The areas that are associated with sensory information occur in the parietal, the insular, the temporal, and the occipital lobes. All right, so basically everywhere but the frontal lobe. And there are 8 main areas that are responsible for sensation, that are associated, I should say, with sensation. So we have the primary somatosensory cortex, which is located here. Somatosensory association cortex. We have some different visual areas, some different, the vestibular areas, which actually are in the insula, so you can't really see them from this particular picture. The olfactory cortex, which you also can't see from this picture because it's kind of located inferior brain area. We have the gustatory cortex, which is also located in this insula, and then the visceral sensory area, which you can't necessarily see from this picture either, but we'll talk about it. So let's start with primary somatosensory cortex. So we already know where this one was primary motor. And then we had that central sulcus, and now we see primary sensory. Okay, and so the primary motor was located in precentral gyrus. We have the central sulcus, and then primary sensory is in the postcentral gyrus of the parietal lobe. The primary somatosensory cortex basically receives general sensory information from the skin and the receptors on skeletal muscle, joints, and tendons, and is capable of what's called spatial discrimination, which basically it means that the primary somatosensory cortex is able to identify which region of the body is stimulating it. Okay, so it's able to interpret the signals and be like, this signal came from your left big toe. This signal came from your right finger. Okay, so it's able to discriminate where in the body the signals came from. Just like the primary motor cortex, the primary somatosensory cortex can also be mapped out to specific areas on the brain. And so on this particular image, in this case, so if this is the front of the head, that makes this the left side, that makes that the right side. And so we are looking at the left primary somatosensory cortex. And what we can see is that there are specific regions of this somatosensory cortex that line up, that receive input to specific areas. So for example, let's say we're talking the gut. This specific area on the primary somatosensory cortex is dedicated to everything gut, everything intra-abdominal. This region of the sensory cortex is dedicated to everything right foot. Right foot. Okay, remember, because we are talking about contralateral. All right, we would have a matching somatosensory cortex over here on the right side, but it would be responsible for everything like left foot, okay? This one specifically, on the left side of the brain, the left hemisphere, is going to map out sensations coming from the right side of the body, all right? With that being said, you'll also notice that some of these things are different sizes. This just basically is representative of the fact that we have many sensory receptors located in the hand and the fingers, and therefore we have a very large area of the cortex dedicated to receiving signals from those regions, where if we look at, say, I don't know, the head, we see only a very small section of the cortex dedicated to receiving sensory information from the head, okay? So secondly is the somatosensory association cortex. And this region is located just behind the primary somatosensory cortex. So this association area, it's like about there. Basically, it integrates sensory input from the primary somatosensory cortex to help you understand an object. So maybe it helps to help you understand the size, the texture, the relationships of the parts of objects that you're actually feeling, okay, and seeing. The visual areas include the primary visual cortex and the visual association area. So we're going to have the primary visual cortex and visual association areas. The primary visual cortex is located, it's in the occipital lobe on the complete posterior tip side, and it's buried in what's called the calcarine sulcus. But basically, the primary visual cortex receives visual information from the retina. Visual association area basically surrounds this primary visual cortex and it gives meaning to what you're seeing. Okay, so it uses past visual experiences to interpret the visual stimuli. And for example, if you look at somebody, the reason that you know who they are is because the visual association area basically puts together the puzzle piece of seeing that person's face and pulling their name and combining them. Okay, so this is a really complex process that really involves the entire posterior half of the cerebral hemispheres. Okay, so the idea of associating things you see with things that you know and are able to like recognize as something that you know what it is because of this visual association area, but it involves many different parts of the cerebral cortex. So clinical connection here. If you have damage to your primary visual cortex, that will lead to blindness. However, if there is damage to the visual association area, the person can still see, like they can see, but they can't comprehend what they're actually looking at. Okay, and so the medical term for this is called visual agnosia. All right, and so this figure here is an MRI, and you can see areas of, you guys might not be able to recognize this, but there's definitely areas here of the visual association area that are completely atrophied. Okay, so this person, this patient, they can still see. Seeing is not a problem. It's just they don't know what they're looking at. So that would be a very difficult thing to kind of wrap your head around if that were you, to constantly not have any idea what you're seeing. The next areas, sensory areas that I'll briefly mention here are the auditory areas. So you have your primary auditory area, and that's located here, and then your auditory association area. Basically, your primary auditory area interprets the information from what you hear, things like pitch, loudness, and location. And then the auditory association area, which is located just behind that primary auditory cortex, its job is to store the memories of sounds, and also sort of permit this perception of sounds. So basically, the auditory association area allows you to, I don't know, recognize songs, for example, as being something that you've heard before, knowing what the sound of a piano is, knowing what the sound of guitar is, knowing the sound of my voice, your mom's voice, so on and so forth, is this auditory association area. The vestibular cortex is also worth mentioning here. It's actually located in the insular lobe, so you'd have to pull the frontal lobe and the temporal lobe out of the way to see the insula. But basically, the vestibular cortex is just responsible for balance and equilibrium. So specifically the position of the head in space, which is the definition of equilibrium. Okay, so it's basically letting you know it's responsible for that standing up and maintaining your balance. Finally, the olfactory cortex. It's olfactory means smell. So you can see the brain has now been cut in half, and now we're looking at, let me just, I'm gonna just make a quick little drawing here. Okay, so if we had our hemispheres here, if this is our left hemisphere and this is our right hemisphere, this picture is if you cut right down the middle of them and you looked at it from the side. Okay, that's what you're seeing here. So the olfactory cortex, olfactory, again, it means smell. This is going to be located on the medial side of the temporal lobe in what are called piriform lobes. But basically, it just is involved in conscious awareness of smells. That's really all there is to it. Additionally, it can also be associated with the limbic system, which is your emotion system. So basically, this is why there are certain smells that can bring up emotional memories for a person. So that's also something that's really interesting, just because of where it's located in the brain. So next we have the gustatory cortex. The gustatory cortex is in the insula lobe. It's just deep to the temporal lobe. And gustatory means taste. And so the gustatory cortex is involved in, it's basically where your taste buds send the signal when you're tasting something. It's going to go to this gustatory cortex and allows you to recognize a taste that you're tasting, whether that be salt, sweet, bitter, umami, so on. Now, this visceral sensory area, it's actually located just behind the taste area. And basically, this particular area, it gives you conscious perception of certain visceral sensations, like an upset stomach or a full bladder. Okay. There's still a lot of research to be done about the visceral sensory area. There's still a lot of stuff that we're not 100% sure on. Okay, so now we've talked about motor areas and we've talked about sensory areas and their related association areas. And so now all that's left is this prefrontal cortex, the anterior association or multimodal association areas. So basically, these multimodal association areas, they get their input from multiple different sensory areas, and then they send outputs out to multiple areas. And so basically, this is kind of what I've been talking about. It's these multimodal association areas allow us to give meaning to the information that we receive by basically storing this information in memories, tying it to previous experiences. And then it also, this is the region where we help decide what to do about the stimuli that we're receiving. So this is also where our sensations, our thoughts, and emotions become conscious. So basically, these areas make us who we are and are broadly divided into three major parts, being the anterior association area, posterior association area, and limbic systems. So I'm just going to highlight those. We have, rather, I'm going to like draw them. Okay. So we have our anterior association area, give or take approximately all of that. We have our posterior association area. Give or take. And we also have this limbic system associative area. Okay, which is going to be the region sort of here. All right. So with that being said, anterior association area. This is also called the prefrontal cortex. Okay, so we've got prefrontal cortex. This is really the most complicated cortical region. This is involved with our intellect, cognition, recall, personality. It contains the working memory that we need for understanding really abstract ideas and judgment and reasoning and planning and persistence. And the development of this area depends on feedback from the social environment.
So next is the posterior association area. And this is the large region that is in the temporal, the parietal, and the occipital lobes. And this particular area is responsible for playing a role in recognizing patterns and faces, and then also localizing this in space. Specifically, there is a region in this area called Wernicke's area, and Wernicke's area is involved in understanding written and also spoken language. Next, we have the limbic association area. And so limbic association area is going to involve the cingulate gyrus, parahippocampal gyrus, and hippocampus. And the limbic association area is going to provide emotional impact that basically makes a scene important to us and helps us to establish memories. So, clinical connection. Tumors or other lesions in the anterior association area might cause things like mental and personality disorders, including loss of judgments, loss of attentiveness, and inhibitions. And so people that have problems with their anterior association area might be oblivious to social restraints, maybe careless about their personal appearance, or they might be big risk takers. But there's different problems that arise for people who have lesions in their posterior association area that provides a sense of awareness of self in the space. So this particular individual might refuse to wash or dress the side of the body that's opposite to the lesion because they might say that region does not belong to me. So they don't recognize things, part of them, as being part of them. Okay, so it's a really bizarre thing to see. And if you've ever watched House, like the show House, they definitely sort of hint at some of these brain lesions. So I've kind of been hinting at this, but basically the cerebral cortex displays lateralization. And lateralization is this division of labor between the hemispheres because the hemispheres, they're not identical. So the left hemisphere typically is the one that controls language, it controls math and logic, whereas the right hemisphere tends to be associated with more visual special skills like painting and artistic type stuff. And also right brain tends to be more associated with intuition and emotion. And so the hemispheres, even though they have different uh, functional capacities in terms of like what they're responsible for, they do communicate almost instantaneously through what we call fiber tracks and also through functional integrations. So kind of going along with this, every person's brain displays cerebral dominance. And this basically refers to the hemisphere that's going to be dominant for language. So remember I said earlier that speech, which is Broca's area, is most of the time located on the left side of the brain, in the left hemisphere. So 90% of humans have left-sided dominance. And this usually results in them being right-handed. So all my right-handed people out there, you are probably left brain dominant, and therefore your speech area is probably located on the left brain. However, in the other 10% of people, the roles of the hemispheres are actually reversed. So in people that are left-handed, all of these things like language and math and logic, and specifically speech, left-handed people have those areas on their right brain. And so it's a really weird phenomenon. If you did like a functional MRI of a left-handed versus a right-handed person, you would see in the right-handed person, the speech area light up in the left hemisphere. But in the left-handed person, you would see the speech area light up on the right hemisphere. And so I found this little cool kind of brain dominance test. So there's the link to it if you want to go and take that test. It's kind of cool. I am actually left-handed. And so when I took this test, and this is probably, I don't know how accurate this test is. It's just something I found online. It's kind of cool. But for myself personally, I actually scored directly in the middle, meaning that both sides of my brain share equal dominance. I'm not dominant one way or the other. I was like dead center 0, which is super bizarre. But I think that also speaks to the fact that we live in a very right-handed world, and left-handed people have to learn how to do things the right-handed way for a lot of instances. And so left-handed people tend to have more equal use of both sides of the brain. So just a fun thing to think about. You can also like do some research on it if this is something that you're really interested in, and share it with the class. That'd be really cool too. Okay, so that takes care of the cerebral cortex. So we're done talking about the cerebral cortex, and now we're going to talk about the cerebral white matter. Okay, so all this white matter that's in all the white matter that's in that region there in the middle. So the cerebral white matter, this is the second of the 3 basic regions of the cerebral hemispheres. And basically the white matter is responsible for communicating between these different cerebral areas, and also communicating between the cortex and the lower central nervous system, so meaning the spinal cord. Basically, again, this white matter is going to consist of myelinated fibers that are bundled into what we call tracts. Okay, so tracts are made of axons, of myelinated axons. And these tracts are classified according to the direction that they run. So association fibers are going to be these horizontal running fibers that connect different parts of the same hemisphere. Okay, so for example, let me get out my pen here. All right, so association fibers. These association fibers, these association fibers, these association fibers. Notice that these fibers, they are starting at one portion and they're ending maybe down there, right? So they're running from one part of the hemisphere to another part of the same hemisphere, okay? Commissural fibers, these are the axons that run between hemispheres. Okay, commissural fibers are these horizontal fibers that are connecting the gray matter of 2 hemispheres. So they are running from one hemisphere to the other hemisphere. Okay, so they would be running here. They are located here because this is basically a half of, this is like one hemisphere. You find the other one sort of
if you could put it back in place. We've cut the brain in half at this particular, so you can see at this particular image. Okay, so commissural fibers, these are the ones that are the axons that are relaying information from left to right hemisphere or right to left hemisphere. Okay. These projection fibers, however, the projection fibers are vertical fibers. And these fibers are going to be the ones that are connecting the hemispheres with the lower brain and also the spinal cord. So there's two sections here of the projection fibers. First is the corona radiata. Basically, these are the projection fibers that radiate through the cerebral white matter of the cortex. And so what I mean by that is, let me just get out a little drawing pen here. So all of this white matter up in here, all of this white matter up in here are all going to be parts of this corona radiata. The internal capsule are the projection fibers on each side of the brain stem that formed a really compact band, and they pass between the thalamus and some of the basal nuclei. So basically, these fibers are going, try to find a color here. Okay, the internal capsule would be all of these, okay, that are, you can see they're crossing over here in the medulla oblongata, all right? And this is why we're getting that crossover of information. So things from the right side of the body go to the left brain, things from the left side of the body go to the right brain. All right, it's because of these projection fibers. Okay. That takes care of cerebral white matter. Super easy piece of cake. Next, we are going to talk about the basal ganglia, or the basal nuclei, sometimes they are called. Just kind of bringing us back to that, these are going to be deep within the white matter. So we're going to talk about what each of these things are. Okay. All right. So each hemisphere's basal nuclei are going to include a caudate nucleus, a putamen, and a globus pallidus. So here we have the putamen and the caudate nucleus, and those two structures together form the striatum. But these particular structures, they're very much so associated with subthalamic nuclei of the diencephalon, and then also the substantia nigra of the midbrain. So here's what you need to know about the basal nuclei. They have a lot of different functions, but some of these functions include muscle movements. They have an influence on muscle movement. Okay, so we influence muscles. Basal nuclei also will play a role in cognition and emotion. They regulate the intensity of either slow movements or stereotype movements. Stereotype movements are things like rapid eye movements. They also filter out incorrect or inappropriate responses so that we don't do them. And then finally, inhibit antagonistic or any unnecessary movements so that we don't perform them if they're not necessary. Okay, so those are the major functions. That's really all you need to know in a sense of the basal nuclei themselves. we're going to talk about these a little bit more later, but just know for now, I'm briefly mentioning that Parkinson's and Huntington's disease are both associated with problems in the basal nuclei. Okay, so each hemisphere has three basic regions: the cortex, the white matter, and basal nuclei, and we've talked about each of them in depth. Okay, so that takes care of cerebral hemispheres. Okay, and here's just a quick little summary table. These summary tables are located all throughout this PowerPoint. They're super helpful. I recommend definitely studying these. They're going to significantly help you on the test. Excuse me. Okay, so we talked about cerebral hemispheres. Now we're going to talk about the diencephalon. The cerebral hemispheres, by the way, that's the biggest portion of this lecture. So things will get a little bit less intense, I think, from here. So the diencephalon actually consists of three paired gray matter structures that all enclose the third ventricle. They're all here in purple.